Hello guys, I am Shalika welcoming you to the show Pharmacy and Medical Crash Course Division in the Knowledge Club online. At previous episode, we learned about basic structure and function of the nervous system, right? Today, I will discuss on the nervous tissue. By the end of this section, you will be able to describe the basic structure of a neuron, identify the different types of neurons on the basis of polarity, list the glial cells of the CNS and describe their function, list the glial cells of the PNS and describe their function. Stay tuned up to end because if you skip the lecture, you can miss our valuable points in between lecture. So stay tuned up to end. I am going to start today's episode. Let's start. Nervous tissue is composed of two types of cells, neurons and glial cells. Neurons are the primary type of cell that most anyone associates with the nervous system. They are responsible for the computation and communication that the nervous system provides. They are electrically active and release chemical signals to target cells. Glial cells or glia are known to play a supporting role for nervous tissue. Ongoing research pursues an expanded role that glial cells might play in signaling. But neurons are still considered the basis of their function. Neurons are important, but without glial support, they would not be able to perform their function. Neurons. Neurons are the cells considered to be the basis of nervous tissue. They are responsible for the electrical signals that communicate information about sensation and that produce movements in response to those stimuli, along with inducing thought process within the brain. An important part of the function of neurons is in their structure or shape. The three-dimensional shape of these cells makes the immense numbers of connections within the nervous system possible. Parts of a neuron. As you learned in the first section, the main part of a neuron is the cell body, which is also known as the soma. The soma is equals to body. The cell body contains the nucleus and most of the major organelles. But what makes neurons special is that they have many extensions of their cell membranes, which are generally referred to as processes. Neurons are usually described as having one and only one axon, a fiber that emerges from the cell body and projects to target cells. That single axon can branch repeatedly to communicate with many target cells. It is the axon that propagates the nerve impulse, which is communicated to one or more cells. The other process of the neuron are dendrites, which receives information from other neurons at specialized areas of contact called synapses. The dendrites are usually highly branched processes, providing locations for other neurons to communicate with the cell body. Information flows through a neuron from the dendrites across the cell body and down the axon. This gives the neuron a polarity, meaning that information flows in this one direction. Where the axon emerges from the cell body, there is a special region referred to as the axon hillock. This is a tapering of the cell body towards the axon fiber. Within the axon hillock, the cytoplasm changes to a solution of limited components called axoplasm. Because the axon hillock represents the beginning of the axon, it is also referred to as the initial segment. Many axons are wrapped by an insulating substance called myelin, which is actually made from glial cells. Myelin acts as insulation much like the plastic or rubber that is used to insulate electrical waves. A key difference between myelin and the insulation on a wire is that there are gaps in the myelin covering on an action. axon, which gap is called a node of Ranvier and is important to the way that electrical signals travel down the axon. The length of the axon between each gap, which is wrapped in myelin, is referred to as axon segment. At the end of the axon is the axon terminal, where there are usually several branches extending toward the target cell, each of which ends in an enlargement called a synaptic end bulb. These bulbs are what make the connection with the target cell at the synapse. So, in gist, the neuronal structure, there are several different types of neurons found in the nervous system. They all contain the same key structural components: the cell body, dendrites, the axon and the axon terminal. The cell body. The cell body holds the nucleus. It is the site of protein synthesis which occurs on small granules of rough endoplasmic reticulum called nuclear substances. In the nervous system, many neuronal cell bodies can group together to form a distinct structure. In the CNS, this is known as a nucleus, and in the PNS as a ganglion. Dendrites. The dendrites are elongated portions of the cell body. They extend towards the receiving input from the environment and from other neurons. Axons. The axon is a long, thin structure down which action potentials, the nerve impulses are conducted. Whilst neurons have many dendrites, most cells only have one axon. Each axon is coated in myelin, a layer of insulating lipid. Myelin is formed by cells wrapping their cells around the nerve axon. In the CNS, it is performed by Oligodendrocyte cells. In the PNS, Swan cells are responsible for this action. There are gaps between the myelin sheaths formed by different cells. These gaps are known as nodes of Ranvier. They allow for salutary conduction of impulses. Axon terminals. The axon terminal is the most distal part of the axon. It is from here that the neuron sends chemical signals to other cells, usually via neurotransmitter release. To facilitate the secretion of neurotransmitters, the axon terminals contains a large number of mitochondria. Types of neurons. There are many neurons in the nervous system, are numbered in the trillions, and there are many different types of neurons. They can be classified by many different criteria. The first way to classify them is by the number of processes attached to the cell body. Using the standard model of neurons, one of these processes is the axon and the rest are dendrites. Because information flows through the neuron from dendrites or cell bodies towards the axon. These names are based on the neuron's polarity. Number 1. Unipolar. Here the cell body is at one end of a single unbranched axon, and there are no dendrites. These can be found in the cochlear nucleus of the brain. Number 2. Pseudo-unipolar. They have one axon which is divided into two branches by the presence of the cell body. Sensory neurons are all pseudo-unipolar. Number 3. Bipolar. These neurons have two processes arising from a central cell body, typically one axon and one dendrite. These cells are found in the retina. Multipolar. They have one axon and many dendrites. With a cell body displaced to one side of the axon, motor neurons are a prime example of this. Then the functional classification. There are three broad functional classification of nerves. That is sensory, the afferent, intermediate and motor, efferent. There are key structural differences between these three types, that is the sensory nerves, small axons and pseudo-unipolar structure, motor nerves, larger axons and multipolar structure, intermediate neurons, central cell body and many dendrites. Sensory and motor nerves are located within the PNS, whereas intermediate nerves are found in the CNS. Now, we will discuss about in detail the neuron cell type in details. Unipolar cells have only one process emerging from the cell. True unipolar cells are only found in invertebrate animals, so the unipolar cells in human are more appropriately called pseudo-unipolar cells. Invertebrate unipolar cells do not have dendrites. Human unipolar cells have an axon that emerges from the cell body, but it splits so that the axon can extend along a very long distance. At one end of the axon are dendrites, and at the other end the axon forms synaptic connections with the target. Unipolar cells are exclusively sensory neurons and have two unique characteristics. First, their dendrites are receiving sensory information, sometimes directly from the stimulus itself. Secondly, the cell bodies of unipolar neurons are always found in ganglia. Sensory reception is a peripheral function. Those dendrites are in the periphery, perhaps in the skin, so the cell body is in the periphery, though closer to the CNS inner ganglia. The axon projects from the dendrite end, past the cell body in ganglion and into the central nervous system. Secondly, the bipolar cells have two processes, which extend from each end of the cell body opposite to each other. One is the axon and one the dendrite. Bipolar cells are not very common. They are found mainly in the olfactory epithelium, where smell stimuli are sensed, and as part of the retina. Number 3, the multipolar neurons are all of the neurons that are not unipolar or bipolar. They have one axon and two or more dendrites, usually many more. With the exception of the unipolar sensory ganglion cells and the two specific bipolar cells mentioned above, all other neurons are multipolar. Some cutting-edge research suggests that certain neurons in the CNS do not conform to the standard model of one and only one axon. Some sources describe a fourth type of neuron called as an axonic neuron. The name suggests that it has no axon and without, but this is not accurate. Axonic neurons are very small, and if you look through a microscope at the standard resolution used in histology, approximately 400x to 1000x total magnification, you will not be able to distinguish any process significantly as an axon or a dendrite. Any of those processes can function as an axon depending on the conditions at any given time. Nevertheless, even if they cannot be easily seen and one specific process is definitely the axon, these neurons have multiple processes and are therefore multipolar. Neurons can also be classified on the basis of where they are found, who found them, what they do, or even what chemicals they use to communicate with each other. For example, a multipolar neuron that has a very important role to play in a part of the brain called the cerebellum is known as a Purkinje, commonly pronounced Purkinje cells. It is named after the anatomist who discovered it. Next one is the glial cell. Glial cells or neuroglia or simply glia are the other type of cell found in nervous tissue. They are considered to be supporting cells and many functions are directed at helping neurons complete their function for communication. The name glia comes from the Greek word that means glue and was coined by the German pathologist Rudolf Virchow, who wrote in 1856. This connective substance, which is in the brain, the spinal cord and spinal sense nerves, is a kind of glue, the neuroglia, in which the nervous elements are planted. Today, research into nervous tissue has shown that there are many deeper roles that these cells play, and research may find much more about them in the future. There are 6 types of glial cells. 4 of them are found in the CNS and 2 are found in the PNS. Glial cell types by location and basic function, that is CNS glia, PNS glia, basic function, astrocyte satellite cell support, oligodendrocyte cells installation, myelination, microglia, immune surveillance and phagocytosis, epidermal cells creating CSF. Glial cells on the CNS, that is one cell providing support to neurons of the CNS, is the astrocyte. So, named because it appears to the star-shaped under the microscope. Astro means star. Astrocytes have many processes extending from their main cell body, not axons or dendrites like neurons, just cell extensions. Those processes extend to interact with neurons, blood vessels, or the connective tissue covering the CNS, that is called the perimeter. Generally, they are supporting cells for the neurons in the central nervous system. Some ways in which they support neurons in the central nervous system are by maintaining the concentrations of the chemicals in the extracellular space, removing excess signaling molecules, reacting to tissue damage and contributing to the blood-brain barrier, that is BBB. The blood-brain barrier is a physiological barrier that keeps many substances that circulate in the rest of the body from getting into the central nervous system, resting what can cross from circulating blood into the CNS. Nutrient molecules such as glucose or amino acids can pass through the BBB, but other molecules cannot. This actually causes problems with drug delivery to the CNS. Pharmaceutical companies are challenged to design drugs that can cross the BBB as well as have an effect on the nervous system. Like a few other parts of the body, the brain has a privileged blood supply. Very little can pass through by diffusion. Most substances that cross the wall of a blood vessel into the CNS must do so through an active transport process. Because of this, only specific types of molecules can enter the CNS. Glucose, the primary energy source, is allowed, as are amino acids. Water and some other small particles like gases and ions can enter, but most everything else cannot, including white blood cells, which are one of the body's main lines of defense, which, while this barrier protects the CNS from exposure to toxic or pathogenic substances, it also keeps out the cells that could protect the brain and spinal cord from disease and damage. The BBB also makes it harder for pharmaceuticals to be developed that can affect the nervous system. Aside from the finding efficacious substances, the means of delivery is also crucial. Also found in CNS tissue is the Oligodendrocyte, sometimes called just oligo, which is the glial cell type that stimulates axons in the CNS. The name means cells of a few branches, that is oligo is equals to few and dendro is equals to branches, the side is equals to cell. There are a few processes that extend from the cell body. Each one reaches out and surrounds an axon to insulate it in myelin. One oligodendrocyte will provide the myelin from multiple axon segments, either for the same axon or for separate axons. Microglia are, as the name implies, smaller than most of the other glial cells. Ongoing research into these cells, although not entirely conclusive, suggests that they may originate as white blood cells called macrophages that become part of the CNS during early development. While their origin is not conclusively determined, their function is related to what macrophages do in the rest of the body. When macrophages encounter diseased or damaged cells in the rest of the body, they ingest and digest those cells or the pathogens that cause disease. Microglia are the cells in the CNS that can do on this in normal, healthy tissue, and they are therefore also referred to as CNS-resistant macrophages. The epidermal cells is a glial cell that filters blood to make cerebrospinal fluid, that is CSF, the fluid that circulates through the CNS. Because of the privileged blood supply inherent in the BBB, the extracellular space in nervous tissue does not easily exchange components with the blood. Ependymal cells line each ventricle, one of four central cavities that are remnant of the hollow center of the neural tube formed during the embryonic development of the brain. The choroid plexus is a specialized structure in the ventricles where ependymal cells come in contact with blood vessels and filter and absorb components of the blood to produce cerebrospinal fluid. Because of this, ependymal cells can be considered a component of the BBP or a place where the BBP breaks down. These glial cells appear similar to epithelial cells, making a single layer of cells with little intracellular space and tight connections between adjacent cells. They also have cilia on their apical surface to help move the CSF through the ventricular space. Glial cells of the PNS. One of the two types of glial cells found in the PNS is the satellite cell. Satellite cells are found in sensory and autonomic ganglia, where they surround the cell bodies of neurons. This accounts for the name based on their appearance under the microscope. They provide support, performing similar functions in the periphery as astrocytes do in the CNS, except, of course, for establishing the BBP. The second type of glial cell is the Swan Cell, which insulate axons with myelin in the periphery. Swan cells are different than oligodendrocytes in that a Swan cell wraps around a portion or only one axon segment and no others. Oligodendrocytes have processes that reach out to multiple axon segments, whereas the entire Swan cell surrounds just one axon segment. The nucleus and cytoplasm of the Swan cell are on the edge of the myelin sheath. Now come to the point, the myelin. The insulation for axons in the nervous system is provided by glial cells, oligodendrocytes in the CNS and Swan cells in the PNS, whereas the manner in which either cell is associated with the axon segment or segment that is insulated is different. The means of myelinating an axon segment is mostly the same in the two situations. Myelin is a lipid-rich sheath that surrounds the axon, and by doing so, that creates a myelin sheath that facilitates the transmission of electrical signals along the axon. The lipids are especially the phospholipids of the glial cell membrane. Myelin, however, is more than just the membrane of the glial cell. It also includes important proteins that are integral to the membrane. Some of the proteins help to hold the layer of the glial cell membrane closely together. The appearance of the myelin sheath can be thought of as similar to the pastry wrapped around a hot dog for pigs in a blanket or a singular foot. The glial cell is wrapped around the axon several times with little to no cytoplasm between the glial cell layers. For oligodendrocytes, the rest of the cell is separate from the myelin sheath as the cell process extends back towards the cell body. A few other processes provide the same insulation for other axon segments in the area. For Swan cell, the outermost layer of the cell membrane contains cytoplasm and the nucleus of the cell as a bulge on one side of the myelin sheath. During development, the glial cell is loosely or incompletely wrapped around the axon. The edges of this loose enclosure extend towards each other, and one end tucks under the other. The inner edge wraps around the axon, creating several layers, and the other edge closes around the outside so that the axon is completely enclosed. Myelin sheaths can extend for 1 or 2 millimeters, depending on the diameter of the axon. Axon diameters can be as small as 1 to 20 micrometers, because a micrometer is 1 divided by 1000 of a millimeter. This means that the length of a myelin sheath can be 100 to 1000 times the diameter of the axon. Revise it. I am damn sure you will require less time to remember all these structures. Write me at the comment box how much you enjoyed and time take to remember the heart anatomy after viewing today's lecture. Next episode, we will discuss on the function of the nervous tissue. Before ending today's episode, if you like this video, please like, share and comment freely. Don't forget to subscribe our channel. If you already subscribed, then thanks a lot. And after subscribing our channel, please click on the bell button for getting the regular updates of Knowledge Club Online. I am coming with another episodes very shortly for you. Till then, goodbye.